Yes.
The next presentation, it will be done by Dr. Muhammad Morhami Ahmad from Malaysia.
Oops.
Okay.
Okay, Dr. Muhammad, you have co-host permissions.
Yeah.
You can unmute and share your
Sure.
screen.
Okay. Muhammad would like to share your presentation, or
I will present by myself.
okay, perfect.
I think
Perfect.
okay, the line is clear, isn't it?
Perfect.
Okay. Okay. Can you see my slide?
Yes.
Yes, perfect.
All right. Okay. All right. Okay. Bismillahirrahmanirrahim. Thank you to the chairperson. Assalamualaikum and a very good evening to all fellow scientists. My name is Muhammad Nohelmi bin Ahmad. I am PhD student from University Malaysia Sarawak, located in the heavenly island of Borneo. Today, I would like to present my research entitled Synergism of Silage Production with Probiotic Reinforcement by Recycled Lactococcus lactis IO1 from the lactic acid fermentation using Sago frond. As a sole representative of Sago Palm, it is my humble honor and my humble opportunity to introduce a little bit more details about Sago Palm. Sago Palm, scientifically known as Metroxylon sago, mainly can be found in Malay Archipelago and Southeast Asia. In Malaysia, 70% of Sago Palm is cultivated in Sarawak. Hence, Sago Palm is one of the major commodity for the state of Sarawak. Sago palm used to be source of staple food for the native community in Sarawak, especially for the production of sago starch. Sago starch used as a main ingredient to make numerous delicacies. For example here, a jelly-like known as linut, and my personal favorite, sago pancake. In this modern era, sago starch used to make noodle. Instead of carbohydrates, sago also provides source of healthy, delicious, and creamy protein in a form of sago worm. Sago palm is very resilient crop that can survive under extreme conditions such as grow at highly acidic soil where most of cash crop cannot survive. And here, the picture shows that sago palms survive from wildfire that frequently happen in Sarawak during hot and dry season. The second picture shows that sago sucker rejuvenate from the same rhizome of sago palm that burn to the ground. Sago palm also can survive against prolonged flood due to its nature that practically live in water that influenced by water type. These advantages are very important for the farmers to recover faster and easier and no need to replant from the catastrophic natural disaster where the earth is rising rapidly nowadays. Due to that, sago play important role as sustainable source of starch in the global market. Sago shows competitive productivity of starch compared to other crop where less land required to produce large amount of starch. Sarawak is the major player of sago starch producer, where we produce 200,000 tons sago starch per year, mainly for domestic use, and export 50,000 tons, mostly to the Asian country, such as Japan and China for the production of food, and some to the United States for industrial application. However, from the graph, we can see that the pattern of sago plantation and starch production were gradually decreasing from time to time. Unfortunately, usually it took 10 to 12 years for the sago palm to be harvestable, and between that time, there is no product that can be obtained from sago palm plantation. This make time is the biggest challenge in sago plantation. Due to that, some sago area was replaced to plant cash crop that makes sago area become less and lesser. On the other hand, our socio-economic study shows that more than 50% of young generation are not willing to continue the legacy due to lack of modernization of sago harvesting practice that make it so difficult to be conducted. As we can see from the picture here, there are two loggers swimming together with the sago log on its way to the nearest sago mill. In some cases, there were reported sago loggers were killed by crocodile while transferring the log. This makes the job not only difficult, but also dangerous. For the last 2 decades, our research team have been struggled to improve sago industry by developing new commodity from the byproducts such as residual sago fiber, that known as hampas, sago effluent as a media for microalgae cultivation, and sago bark for decoration product, without affecting the production of the starch. But still, we have to wait for the sago palm to be harvestable. Okay, those are post-harvest. We still need to harvest the sago palm, then we can have this byproduct. So in our journey to enrich the sago industry to research and development, we were inspired by strong and dependable women entrepreneurs that empower the socio-economic of the household through creativity of using sago leaf and sago frond to make this beautiful and authentic roof of this hut and those beautiful handcrafts. From there, we discovered the ecological sustainable of sago frond to be industrially utilized as a raw material for the production of high value product. We projected that we can obtain 40 tons of sago frond from 1 hectare of sago farm. Most importantly, new frond will grow every month so the supply keep on coming. As we can see from here, the pruning of frond from young palm is commonly practiced by the farmers to stimulate the growth of the mature and older palm that lead to massive accumulation of sago frond abundant in the farm. The aim of this study is to develop alternative commodity from sago palm plantation as early as 2.5 years, then to produce sustainable animal feed from locally available resources and improve the quality of the feed with probiotic supplementation. From that, we expect enhancement of sago palm plantation and productivity so we can maintain the starch supply to the global market. We projected this research will stimulate livestock industry development by reducing dependency on imported feed and beef by developing locally available resources to produce feed. Currently, Sarawak self-sufficient level for beef is only 12% and mutton only 5.5%, while we expected to increase the self-sufficient level for both at 50% by 2030. On top of that, Sarawak have to import 430 million ringgit worth of animal feed every year to maintain the current productivity of our livestock industry. Hence, this research will support the vision of Sarawak state to become the net food exporter by 2030. The schematic diagram shows the subsequent process flow of lactic acid and silage production. It begins with the extraction of sago frond sap simply by using roller pressure machine, as shown in the picture here, and the sap was formulated as medium to produce lactic acid and to grow IO1 cell mass. Meanwhile, the residual rachis and leaf were collected as raw material for the production of silage. Sago frond sap processes glucose and xylose as major sugar content associated with traceable amount of enzyme cofactor that able to stimulate the growth of IO1. Boiling at 100 degrees Celsius is sufficient to sterilize the sap for pre-treatment before the fermentation begins. We took advantage of the heat energy from the boiling to conduct simultaneous enzymatic hydrolysis to convert the residual starch to glucose to maximize the sugar content. We formulate the fermentation media with different dilution factors to determine the most efficient lactic acid and IO1 production. Figure shows the density of IO1 cultured in MRS agar isolated from media with different sap concentration. From the result, 50% diluted or 35 grams per liter sugar content, sap media exhibit the most efficient production of lactic acid together with lactic acid Lactococcus lactis IO1 production. This was associated with the most effective sugar consumption. That means we do not waste the sugar. In formulation, with sago frond sap increase the performance of Lactococcus lactis IO1 higher than the normal or the general Lactococcus lactis using the standard media. Using the standard media, they are just around 20 grams per liter. But we improve the formulation so it can digest higher sugar concentration, which is 35 grams per liter. From there, we cultivate Lactococcus lactis IO1 using the optimized formulation of Sago frond sap media and recover the cell from the production of inoculant that we patent as Silage for the production of Sago frond silage. After considering the factor of dry matter, digestibility of the fiber, total water soluble sugar, minimum pH and protein content, we conclude that 1:1 ratio of leaf and residual fiber from the previous method. We collect it again and we mixture it 1:1 ratio, produce the ideal condition for the production of large-scale silage. In order to determine the efficiency of Silage, we studied the effect of inoculation of Lactococcus lactis I01 on the production of Sago frond silage. As you can see from the first graph, Silage managed to improve the acidification during ensiling process. This is very important because we need to acidify the silage as fast as possible because we need to inhibit the growth of the pathogen, the growth of the fungus, the growth of bad bacteria. So we need to do it fast. So Silage that we include in the silage help us to do it faster and better. But for the pH, however, the pH increase again after 3 weeks of ensiling. That will be explained in the second graph. The second graph shows the accumulation of lactic acid and acetic acid associated to the consumption of sugar in the silage. The accumulation of lactic acid and acetic acid in non-inoculated silage rarely increase up to 4 weeks of ensiling. However, inoculated silage with Silage show drastic increase only the first 2 weeks, but after that it start to decrease again. This is because of the acidity in the silage itself trigger lactic acid degrading bacteria to metabolize the lactic acid into acetic acid to control the acidity of the silage. This mechanism is important not only to prevent the growth of undesired microorganisms, but also to maintain the ideal condition of the probiotic to remain viable in the silage. On top of that, this will allow us to introduce and populate the probiotic into the rumen ecosystem of the animal model through the silage. So when the animal model consume the silage, the probiotic will survive and inside the body and inside the digestive system. And we hope our further study that we will find that I01 survive in the manure of the animal model so that we can improve the manure as an inoculum or as a nutrient into the production of fertilizer.
We have again 4 minutes maximum, please.
Okay. All right. I have another just a few. The success of Silage inoculation was reflected through ability to preserve valuable nutrient content and preservation of secondary contamination from undesired microorganisms. As you can see from the first graph, we managed to reduce protein degradation. Without the Silage, it reduced 13%, but with the Silage, it only reduced to 6%. The second graph show the contamination after we expose the silage to the air. Okay, as we can see, the lactic acid bacteria keep on growing even after we expose to the air, because I01 categorized as facultative bacteria that can grow with or without oxygen. For yeast, yeast in inoculated silage only reach the threshold level, which is the maximum level, after 8 days, after 4 days of exposed to the air. But for the non-inoculated, it grows faster. But in some study, that shows that yeast actually improve the growth performance of the ruminant. So it doesn't matter. Our very most concern is on the growth of the mold here. As you can see from the graph here, mold was identified at the day 0 of non-inoculated silage. This means that the mold already manifests the silage during ensiling process, which is the conventional method is not effective. So when we add Silage into the silage, it improved. It managed to inhibit the growth of the mold during ensiling process. And as you can see from the graph here, the mold remain under threshold level even after 12 days of exposure to the air. This is very important. If we manage to inhibit the growth of the mold, we manage to prevent the production of aflatoxin that can compromise the safety of the silage for ruminant consumption. to show the effect of the ensiling. Actually, the ensiling process improve the efficiency of the silage itself, because the animal model, in this case, we use Malin breed sheep. They consume the same amount of feed, but the average daily gain is higher instead of commercial pellet. We compare it with commercial pellet. Okay, and we conduct the feed efficiency. That means here, if I have 10 kilogram of commercial pellet, I only can give to 10 sheep in one day. But if I have 10 kilogram of Sago from silage with inoculation, I can feed 15 to 17 sheep in one day. So inoculation and usage of silage, usage of Sago frond is improving the efficiency of the silage, of the feed itself. So we postulate the feed cost to produce the sheep using inoculated Sago from silage was around 280 ringgit only, compared to 540 ringgit by using commercial pellet. This is starting from growing stage of the sheep until it can be slaughtered. Okay, so this is the cost. So by using silage, we cut 50%, almost 50% of the feeding cost, which is involving 70% of the total cost to produce sheep. So for the conclusion, usage of Sago frond as raw material, providing ecologically and economically sustainable resources for the production of animal feed. Through this work, we managed to improve the quality of the silage with Lactococcus lactis IO1 inoculation, providing the livestock with healthier and safer feed. The enrichment of byproduct from Sago palm to produce high-value product will empower Sago industry through technology and knowledge transfer. We believe Sago palm is one of the key to the future of global food security and safety. Best regard from my research team. This project was fully funded by Fundamental Research Grant by Ministry of Science Malaysia. I think that's all from me. Thank you for your attention. Assalamualaikum.
Dr. Badawi, unmute yourself. Nice presentation. Thank you.
Thank you. Thank you, Prof.
Thank you.
Thank you, Mr. Mohammed. It is really very good presentation. I think we don't have time for question, or Dr. Ramzi?
If there is one question, it can be asked to Dr. Mohammed with pleasure.
Do we have a question?
If now